Hey scientists, so yeah, wearing the safety goggles today. Never can be too safe when you're talking about cell chemistry, part 2. So today we're going to be chatting about the most important organic molecules. Remember, organic has carbon. So there are 4 types: carbohydrates, lipids, proteins, and nucleic acids. So let's go into a little more depth with each one. Let's start with carbohydrates. All right, so carbohydrates are basically sugars. They are used for shorter-term energy storage. So this is the first form of energy that's burned. So as soon as your body needs energy, it's going to go to carbose first. There are three types of carbohydrates. The first are monosaccharides. The second are disaccharides. And the third are polysaccharides. So let's go into those in a little more detail. All right, so monosaccharides are just simple sugars, so not very complex. They are the monomer of carbohydrates. And remember, monomer is a single. So again, it is used for quick energy retrieval. So the chemical formula is usually C6H12O6. So 6 carbons, 12 hydrogens, and 6 oxygens. All right, some examples of monosaccharides. Glucose. So that is basically blood sugar. So, oh, my glucose level's low. That is the sugar that exists in your blood, in your bloodstream, in the form of blood sugar. Fructose, which is fruit sugar. So you're eating an apple, that is fructose. So from fruit, generally speaking. All right, disaccharides. Now, remember we're getting more complex now. Now we have 2 linked monosaccharides. So you're getting the monos and you're making the polymers. So you have the monomers and you've got the polymers, remember from part 1. So now we have 2 linked monosaccharides. So this is called a carb polymer. Remember, poly is many. So this is also used for short-term energy storage. So some examples of this might include lactose, which is sugar that is found in milk, sucrose, which is just straight up cane sugar, and maltose, which can be found in wheat. All right, polysaccharides. These are 3 or more sugars that are linked. So again, poly is many. So as you see, as we're going a bit down a bit further, they get a little bit more complex. The structure gets to be a little more complex. All right, again, still a carb polymer meaning many. So this is used for long-term energy storage and also for structural support in the cells. So this is, if you eat too many, this is often what can be converted to fat. So if your body does not end up burning through these polysaccharides, it goes, hey man, I'll just kind of go ahead and just bank it over here a little bit, just in case I need energy for later. So these polysaccharides are most often the ones that are converted into fats. All right, some examples of polysaccharides. Starch. This is plant energy storage, like potatoes, things like that. Cellulose. Now the cellulose is what plants use. Remember what I said up here that it is used for structural support. So right here, structural support. So what that is cellulose, which is what plants use to sort of give themselves structure, sort of plant bones, so to speak. All right, and glycogen, which is fuel for endurance sports, so marathoners, things like that. People or the athletes that need sort of more longer energy stores is glycogen, which is a polysaccharide. All right, lipids: fats, oils, waxes, and cholesterol. Now, fats take a longer time to break down, so they are used for longer-term energy storage. Actually, fat has the most energy in it. It can store the most E. Think of it sort of like an E-bank. It's sort of where you store the energy for later. So like you eat breakfast, and then you're like, okay, so I want to bank that out a little bit, so that as the day goes on, you have energy to get you all the way through the day. That's why you always need a little bit of fat in your diet. All right, fat does not dissolve in water, and we will get a little bit more into that a little bit down the road. It's not water soluble. All right, so types of lipids. So basically, fats are lipids that are solid at room temperature. So big old glob, room temperature, bam, they're going to be solid, okay? Now, saturated. all possible H is full. Now, H meaning hydrogen. So the thing about fats is it's all about the hydrogen and the way the hydrogen bonds. So this is going to be full of energy. And it's going to be stiff, which is why it's solid at room temperature. All right, so the difference between fats and oils is oils are lipids that are not solid at room temperature. These are unsaturated, meaning that they do not have all of their hydrogen is not full. All the hydrogen links are not full. This is a little bit floppy, so the molecule is going to be kind of a little bit floppy versus the fat molecule, which is going to be more solid. All right, fats and oils. So again, like I said, it is all about the hydrogen. If you will look here, you will see that there is lots and lots of hydrogen atoms. So let me go ahead and draw here. So we've got saturated fats up here. Hydrogen, 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 hydrogen. Okay? This is a saturated fat. Now, this is an unsaturated fat. Let me go ahead and click off that, because that's really annoying. Okay, stay with me here just a second. Okay. I'm going to dismiss this and ignore forever, because you just totally... All right. So hang with me here. Going to get back to it. All right. Thank you for that little detour. The exciting things that happen when a computer takes over for itself. So anyway, where were we? I think we were red here. Yeah. So, okay. So if you've got lots and lots of hydrogen here, right? Now, if you will kind of boogie with me down here to unsaturated fat right here, you will notice that there are still links here that can still accept hydrogen. All right. The big difference between a saturated fat and an unsaturated fat is that basically the hydrogen has almost all of them, all of the links occupied. I mean, yep, there still are a few right here, right down here, but basically it is just chock full of hydrogen, okay? Big difference between saturated and unsaturated fat. All right, so a lipid structure. There's a triglyceride, glycerol head, which has 3 fatty acid tails. So tri, remember, like tricycle, triplane, 3. Tri is 3. 3 heads and 3 tails. This is a monomer of a lipid. Okay, monomer meaning 1. Now, the glycerol head, this is important. So the glycerol head is hydrophilic, okay, which means that it is water-loving. I love water. Okay. Now, as you would imagine, the fatty acid tails are hydrophobic. Okay, so hydrophobic is that they have a fear, a phobia of water. So the tails are hydrophobic, and the heads are hydrophilic. Keep that in mind. It's going to come in handy. All right. So phospholipids, lipids that make up the cell membrane. Now, it has 2 tails and a phosphate attached to glycerol. So the cell membrane is made of a double layer of these guys. It's a lipid bilayer, bi meaning 2. Now, this is something that helps transport through the cellular membrane. So nutrients like to attach to the hydrophilic head, but not to the hydrophobic tails. So essentially, the nutrients come in and attach to the head, but not to the tail. All right, so protein, or organic molecules that are used to build body parts. These are made up of amino acids. Now, an amino acid are the building blocks of cells. This is a monomer of a protein. There's basically 20 common amino acids. So these sort of act like letters of the alphabet. There's different combos that make different proteins. So a peptide bond is a bond between the amino acids. It takes heat to separate these apart, to break these bonds. All right, an enzyme. An enzyme is protein that aids in chemical reactions. Remember we were talking about the enzymes in part 1. So this can make a reaction start or stop, kind of like an on and off switch. So it needs less energy to start a reaction. So it can break down food or build new muscles, etc. So enzymes do a ton of things. They build muscle. They help move matter and sort of stuff around the cells. Enzymes are actually really, really important. So remember, this is remember how we were talking about hydrolysis, the two types, hydrolysis and dehydration synthesis. All right, so the way enzymes work, it's basically a lock and key model. So each enzyme is shaped to break down a specific type of protein. So each enzyme can only act on one substrate. So each enzyme will only work with one thing. So it has to be the right shape for the enzyme to lock onto the substrate. So if you look here, this enzyme will only work with this substrate. Okay, so it's the lock and key. So it will only fit in here. And again with these guys down here. And remember, it starts and stops a process. All right, so the substrate is the substance to be broken down or put together. Nucleic acid. These are the building blocks of DNA. So these contain all the instructions necessary for a cell to live and reproduce. So the nucleotide is the monomer of a nucleic acid. Getting the pattern here, another monomer, meaning one. All right, so there are two types of nucleic acids, DNA, and the other one is obviously going to be RNA. So let's look really briefly at DNA structure. All right, so basically DNA is like a spiral staircase. A always pairs with T, and G always pairs with C. So if you look down here, these guys will only pair together. A and T, G and C. That is the only way that it's like lock and key. It'll only mix with certain things, and it wants to hang out with certain people, certain kids. All right. So the double helix model is basically the spiral shape of the DNA. And again, we're going to go over DNA much more in depth in class. All right. So cellular chemistry. So this is going to be something that we're going to work on in class. We'll go over what the organic compound is, what the monomer of it is, and the role that it plays in the cell. So that is cell chemistry part 2. Take out my safety goggles, and I'll see you in class.